Good evening, everyone. Good evening, and welcome to yet another supremely amazing lecture on the chapter Microbes in Human Welfare, which is a brand new chapter we are discussing. So, this chapter, or in this today's class, we'll be discussing about two main topics. That is, we'll be discussing with microbes in industrial products and microbes in household activities. So this is your lecture number 1 on the very brand new chapter, that is microbes in human welfare. Those who haven't yet subscribed our wonderful YouTube channel, don't forget to subscribe BioPoint NEET, stream the world of bio. This is our amazing Telegram group at BioPoint New where we conduct regular quizzes, polls, updates on our classes, OMR examinations and daily questions will be done over in this very small but informative Telegram group. So we are starting with this chapter, Microbes in Human Welfare, which is our chapter. Whenever we hear of the term microbes, the thing that comes to our mind is they cause diseases. But we have to understand, first thing that we have to understand is that microbes are not only the organism that causes diseases. What does the term microbe refers to? What does the term microbe refers to? Microbe means they cannot be seen. They cannot be seen with the help of our naked eyes. That is actually the meaning. But we have all misunderstood the meaning of microbes as the substances or the organism that causes diseases. Am I right? So in this chapter, we are going to discuss about some of the useful aspect or the useful areas, some of the areas where the bacteria, fungi, protozoan, virus, all these finds their utility. So this chapter mainly divided into 6 main topics, that is microbes as household products, microbes in industrial products, microbes in sewage treatment, microbes in biogas production, microbes as biocontrol agents, and last one, microbes as biofertilizer. So 1 and 2 topics included in our lecture number 1, sewage treatment and biogas production included in the lecture number 2, and biocontrol agents and biofertilizer included in the lecture number 3, which will be followed by the MCQ section. Yes, these are some of the varieties of microbes that are meant in your text. Microbes that here in this diagram we are referring with the bacteria. What do you mean by bacteria? Bacteria are actually prokaryotic species, right? Am I right? Prokaryotic species, and they are included. So prokaryotic means they are included under the kingdom Monera, because Monerans is the only kingdom of the 5 kingdom proposed by R.H. Whittaker that has a prokaryotic nuclear association. So they can be divided in the second chapter of class 11, that is biological classification. We studied that the bacteria can be divided into 4 based upon their shape. Am I right? So the rod-shaped bacteria, what you will call it as? The rod-shaped bacteria, you will call it as bacilli. Next, spherical-shaped bacteria, you will call it as coccus. Here again, they are showing a rod-shaped bacteria which is having a flagellum. What is the function of flagellum? Flagella helps in the locomotion, right? So these are some of the images which is mentioned there in your NCERT itself. Next, very important one, bacteriophage. What do you mean by a bacteriophage? It is not a bacteria, but it is actually a virus. So bacteriophage means the name suggests it is not a bacteria, but it is a virus. The virus that attacks bacteria, you will call it as. So bacteriophage are those viruses that attacks bacteria. Okay, bacteriophage are those viruses that attacks bacteria. Next you have, so this is an important diagram. I'm giving you 110% guarantee that this can be a diagram based question in the upcoming NEET examinations. So you have the tetragonal or the hexagonal shaped head, then you have the collar followed by the tail. These are the three main parts which can be asked for your exam. Next, the second diagram, that is this particular diagram, shows the adenovirus and what is its cause. It causes certain respiratory infections. And we have the rod-shaped tobacco mosaic virus. So from all these things, I have been constantly telling that these causes diseases, right? So in this chapter, we are going to study about certain microbes which are useful to human beings. So this is a major doubt which I'm going to clear right now. That is, this is how certain colonies of bacteria or fungi are produced during the experiments. When we are conducting an experiment, accidental discovery of penicillin by due to the formation of the fungal growth on a petri dish, right? So this is how the colonies of bacteria or fungi just grows on the petri dish. Okay. Next one, that is, in this chapter we are going to discuss or move into detail of this chapter. That is the first topic of this chapter, none other than microbes in household product. In the household items, in which all items are we using the microbes? So let's have a look at the very first topic. That is, the first thing is none other than, it is about the Lactobacillus bacteria, or you can call it as the lab. Very, very important abbreviation question that can come for your NEET exam. Lab. What is lab? What is lab? Lactic acid bacteria, which can also be called as the lactobacillus bacteria. What is the lactobacillus bacteria? It converts milk into curd. It converts the milk into curd. As the name suggests, bacillus. Look at the shape of the virus. They are rod shaped, right? They are rod shaped bacteria, so that's why they are called, and they are concerned with the lactic acid formation, right? So hence they are called lactobacillus bacteria. So it converts milk into curd by producing certain acids that helps in the coagulation and partial digestion of the milk protein. So these bacteria will produce certain kinds of acid inside their body, which they helps in, which they makes or which they utilize to make it coagulated or partially digest the milk proteins. What is the milk protein? Common milk protein, you can call it as the casein. Casein is the common milk protein. Next, fresh milk. The fresh milk can be converted into curd. You might have seen in your homes, your mother might be adding a small amount of curd into the fresh milk or the milk which have to be turned into curd by the morning or the daybreak of the next day. So that small amount of curd that you are adding into the fresh milk, you will call it as the inoculum. What you will call it as? You will call it as the inoculum. Inoculum means that contains millions of bacteria, millions of this lactobacillus bacteria. That's why it helps in the easier conversion of milk into curd. And one more important function of this particular bacteria, it increases the vitamin B12 content. It increases the vitamin B12 content of the curd. Another important function, the lactobacillus bacteria plays an important role in our stomach by checking out the pathogens. So it acts as a checking mechanism for the pathogens that enters our stomach. Next is bacterial fermentation, anaerobic respiration. What do you mean by actually fermentation? Fermentation means it is commonly known as anaerobic respiration, and this particular bacteria or the microbes are you not only bacteria. We use microbes making food items such as dosa and idli, etc. And how the puffed appearance of the dough, that is when you are making the dough for preparing your dosa or idli or whatever or any other food items, you may have the puffed up appearance of the dough, and that is due to the production of a gas, that is the carbon dioxide gas. At least 20 times repeated MCQ for your NEET. Which evolution of which of the following gases causes the puffed appearance of bread or dosa or idli or whatever? You will have the carbon dioxide. And from where is this carbon dioxide coming inside this food? By the bacteria. The releases of the bacteria releases the carbon dioxide, right? So that is another important area where you are using the microbes. Next, one of the important one is the baker's yeast. Baker's yeast, another the scientific term is Saccharomyces cerevisiae. Very important. This is very important. This is the only kind of species coming under Saccharomyces, that is cerevisiae, which can be called as the baker's yeast. What do you mean by baker's yeast? It is used to make bread by fermenting the dough. By fermenting the dough or the substance that we have used for making the bread, so that the bread, you have the puffed appearance. Yes, you have the puffed appearance. The puffed appearance is due to the particular kind of fungus which is known as Saccharomyces cerevisiae, baker's yeast. So I'm giving you a homework. You have to tell me the class whether yeast belongs to Ascomyces, Phycomyces, Basidiomyces or Deuteriomyces on the comment section after this class, which will be open to you after this class. Okay, you have to comment me there on the public comment section. Which class of fungi? We studied different classes of fungi, 4 different classes of fungi in your 11th standard. So among that 4 different, yeast belongs to which class of fungi? That's my homework and you have to comment me the answer. Next household product, you can see the action of microbe is the toddy. What is toddy? Toddy is actually different from alcohol, but both have an alcohol content. It is made by fermenting the sap from the palm trees. You might have seen this. Okay, so the microbes are used to ferment the fish, soya bean, bamboo shoots, and to produce cheese. Very, very important, guys. Very important. You have to study about the 2 types of microbes that causes the formation or that helps in the formation of cheese. Clear? Let's move with the formation of cheese. So in this chapter, according to your syllabus, you have to study 2 different cheese, that is the Swiss cheese and the Roquefort cheese. What are the 2 kinds of cheese? Swiss cheese and the Roquefort cheese are the 2 different kinds of cheese that you have to study in this chapter. First, we are going to discuss about the Swiss cheese. Swiss cheese means they have large holes. If you can, after the class, you can go to Google and you can search out the image. Swiss cheese and the Roquefort cheese. Swiss cheese means they have large holes in them and that are produced due to the production of carbon dioxide gas by a particular type of bacterium which is called the Propionibacterium shermani. Very important MCQ. We have to expect this question in the section of match the following. Very, very important. Propionibacterium shermani. Very important question. Then you have the Roquefort cheese. It is ripened by growing a fungus that is Penicillium roquefort. Penicillium roquefort on them. So another second homework that I'm giving you 2 homework. First one, to identify the class of fungi in which you can find out yeast. First homework. Second homework, the class of fungi where you can see penicillin. Separately, you have to tell me yeast, put a hyphen, tell me the class. Penicillin, put me what? Put a slash or a hyphen and tell me the answer for that also on the comment section. So Swiss cheese by Propionibacterium shermani and the Roquefort cheese by Penicillium roqueforti. Yes. Next, we are moving on to yet another section of this chapter, that is microbes in industrial products. What? industrial products we are going to discuss. Yes. So first we are going to next the second part of this chapter, that is the industrial products. Production of beverages, antibiotics on an industrial scale requires the growing of microbes on large vessels called fermenters. This is an important MCQ which was asked for a previous year JIPMA paper. Okay, the very large vessels in which we grow microbes for industrial purposes, you will call it as fermentators or fermenters. Okay, so these are the large fermenters in which you have been growing the microbes at particular temperatures. Next, first we have to discuss about where all are the microbes used in the fermented beverages. Okay, Saccharomyces cerevisiae, which is now known as the brewer's yeast also. So yeast, two names are there. In household products, we call it as the baker's yeast because they are used in the production of bread. In industrial activity, you will call it as the brewer's yeast because they are used in the production of beverages. This beverages is again produced by the material or the organism which is known as yeast, which is a fungus, by fermenting malted cereals. What do you mean by malted cereal? This is the malted cereals. Okay, by fermenting the malted cereals or the fruit juices to produce a particular thing, that is ethanol. Guys, too much amount of ethanol or methanol or whatever alcohol is harmful to our body. Next, you have two different kinds. These all are the common brands of alcohols that you might have heard. Suppose you might have read it in the newspaper. Suppose you might have heard out in some films or whatever. Okay, so these are some of the brands of alcohol that we have to study or that are mentioned in our NCRT textbook for you to refer. First is wine and beer. Wine and beer are the alcohol which are produced without distillation. These 2 points are very important. So you have to study wine and beer. They are produced without distillation. Whiskey, brandy, rum, gin, arrack, all these things are produced by distillation of fermented broth. So this you have to study what are the different kinds of brands of alcohol that are formed by distillation and that are not formed by distillation. Suppose a question may come for your exam. Actually, this kind of a question asked in 2002 or 2001 for AIMS paper. You have to imagine for AIMS paper, they asked a question like this. Which of the following is not produced by distillation method? So the answer could either be wine or the beer depending upon the options given over there. Let's move on to the next slide. Yes, we have to study about common thing, antibiotics. We all are here to take up MPBS, right? So this is important. Antibiotics. What do you mean by antibiotics? They are actually the chemical substances which are produced by some microbes and can kill or retard the growth of pathogens. So antibiotics are certain chemical substances which are produced by microorganisms and that are used to kill or inhibit the growth of other microorganisms. So antibiotics are used in the treatment of plague, whooping cough, or diphtheria or leprosy or whatever diseases. So antibiotics, which is again an industrial made products from another kind of a microorganism. Another kind of a microorganisms only we are making out this antibiotic. Right? Very important one, familiar thing, penicillin. Penicillin, those who are preparing for your CBSE board 2021, this was a CBSE board 2020 question. Okay, about the Alexander Fleming and his discovery that leads to penicillin. 3 marks question. So Penicillin was the first antibiotic which was discovered by the famous scientist known as Alexander Fleming. He observed that Staphylococcus bacteria could not. I remember right now I show you the petri dish first. Okay. So he was doing certain experiments, right? So in these experiments, actually penicillin discovery or antibiotic discovery was an accidental one. She observed that the Staphylococcus bacteria could not be able to grow on certain plates which are unwashed. You know that if the food item is kept for a long time, if the bread is kept for a long time, definitely bread mold appears on it. Definitely right like that. He observed that certain kinds of fungus was growing on these unwashed dishes or this unwashed culture plates. Certain kinds of bacteria couldn't grow. That is, Staphylococcus was not able to grow in that particular culture plate because due to the presence of a particular types of fungus. So from them he extracted the antibiotic known as penicillin, penicillin notatum. And before that, I have to make understand in the chapter number 1 we studied that disease conidia. Conidia which are actually exogenous in origin, Next is the persons that have been credited with the discovery of antibiotic was Ernest Chain. Guys, Ernest actually spelling you can give it as ERNST according to your NCRT. Ernest Chain and Howard Florey established its full potential as an effective antibiotic. Alexander Fleming was able to discover, but he was not able to put forth all the varieties, all the applicable areas of this penicillin. But Howard Florey and Ernest Chain worked on that and find out the full potential and used it as an effective antibiotic. Next, you have the Fleming, Chain, and the Howard Florey were awarded the Nobel Prize for the discovery of antibiotics in the year 1945. So guys, the name of the scientist is an important question for your NEET exam. So whenever you hear the term Florey, Chain and all these things, you have to remember they are discovering penicillin or they are associated with the discovery of penicillin. 1 second guys, let me check. Yes, we have 2-3 slides more. Next, microbes are used in certain chemicals, enzymes and other bioactive molecules. First one is they are used in the formation of certain organic acids. Acid producer microbes include Aspergillus niger, Acetobacter aceti, Clostridium botulism, Lactobacillus bacteria. First one is the Aspergillus niger. It leads to the formation of citric acid. So Aspergillus niger, it is a fungus. So 3 kinds of question. What is the fungus that produces citric acid? Your answer should be Aspergillus niger. Aspergillus niger that produces a citric acid is which kind of a microbe? It is a fungus. Aspergillus niger produces what? It produces the organic acid named the citric acid. Guys, this is a diagram for Aspergillus niger. Next, you have Acetobacter aceti, which is actually a bacterium that produces acetic acid. This is the diagram for Acetobacter. Clostridium botulism, which causes a disease called what? Canned disease or botulism. Okay, botulism. It's also produces useful by producing a particular kinds of organic acids called butyric acid. Lactobacillus is again a bacteria that produces what? Lactobacillus, or what? It produces lactic acid, as I've already mentioned. Yes, we have alcohol. Second one is the alcohol. So yeast, Saccharomyces cerevisiae used to produce ethanol, which is a kind of an alcohol, right? Then you have certain enzymes. First one is the lipase. Lipase means what? They are used in the detergent formulation. They are used in the detergent formulation, helps to remove oily stains from laundry. Previous year NEET MCQ question. At least 25 times repeated MCQ question coming from the upcoming slides. First one, lipase used in detergent formulation or used to remove oily stains from laundry. Second one, you have pectinases and proteases which are used to clarify the bottled juices. You might have seen whenever you make a fruit juice at your home and the fruit juice that you get from a shop, these two differs very greatly. Am I right? Give me hearts on the screen then. Give me hearts on the screen then. They differ very greatly. Am I right? That is because in the bakeries, in the juices, they will add what? Certain kinds of an enzyme called proteases and pectinases. And for what? They are used to clarify the bottled juices. They are used to clarify the bottled juices. Next one is a streptokinase. We have studied about this in the chapter body fluids and circulation. I already taught you that chapter. Streptokinase, which is produced by streptococcus, names such as coccus. Guys, you have to connect it like this. Streptococcus. Look, they are spherical. They are used as a clot buster. So say, name the substance which is used as a clot buster. It is streptokinase that is used to remove clots from the blood vessels of the patient who suffered a myocardial infarction. Right? So streptokinase, which is produced by a bacterium known as streptococcus, is called as the clot buster bacteria. Next, you have another important bioactive molecule, that is cyclosporine A, which is produced from a fungus, Trichoderma polysporum. MCQ question 50 times. They are used as an immunosuppressive agent during organ transplant. Guys, when suppose a kidney has to be transplanted and I have donated a kidney to one of you and you have received that. So along with the operation, you might be accompanied by giving a cyclosporine molecules also inside your body because they act as a immunosuppressive agent, because at this particular organ transplantation surgeries, our immune system has to get suppressed. Right? The last one is the statin. Statin is produced by an yeast which is again a fungus, Monascus purpurus. Guys, streptokinase, lipase, cyclosporin, statin. 4 terms, very important. So the statin is used as a blood cholesterol lowering agent. So you have the lipase removing oily stains from clothes. Then you have the streptokinase which act as a clot buster, cyclosporin immunosuppressive agent, statin which is used as a blood cholesterol lowering agent. And how they act as a blood cholesterol lowering agent, those who are preparing for your board might receive this question for 1 mark. They are responsible for the synthesis of cholesterol. They inhibits the enzymes. They inhibits the enzymes that are responsible for the synthesis of cholesterol. So in the next class, we will be studying about this particular topic, that is microbes in sewage treatment. So I hope you have enjoyed the class. Don't forget to hit the like button right now. I hope there are no doubts for you. If you have any doubt, you're free to ask me in my Telegram. Okay? Thank you guys. Don't forget to like, share and subscribe our wonderful channel BioPoint Stream, the world of bio. And one request, comment the homeworks on the public comment section after the class. Soon after this, you have to comment. Bye bye guys. Take care.